The lethal dose 50 of botulinum toxin, or BONT, is estimated at just 1 nanogram per kilogram when administered intravenously, making it the most potent known biological substance. Produced by the anaerobic bacterium Clostridium botulinum, this neurotoxin is a large protein complex. The active component is a 150 kilodalton dichain polypeptide. This structure consists of a heavy chain, approximately 100 kilodaltons, and a light chain, 50 kilodaltons, linked by a single disulfide bond. The heavy chain dictates the toxin's binding specificity and facilitates translocation across the cell membrane. The light chain, however, is the functional enzyme, a zinc-dependent endopeptide responsible for cleaving crucial proteins within the neuron. Understanding this bipartite structure is the key to deciphering its paralyzing power. BONT exhibits high tropism for cholinergic nerve terminals, specifically targeting the presynaptic membrane of the neuromuscular junction. The heavy chain contains a receptor binding domain that recognizes specific polysialogangliosides and synaptic vesicle protein 2, or SV2, receptors on the neuronal surface. This high-affinity binding is the first step in the intoxication process, ensuring the toxin concentrates precisely where acetylcholine release occurs. Following binding, the neuron internalizes the toxin complex through receptor-mediated endocytosis, encapsulating it within an acidic vesicle. This precise targeting mechanism explains why systemic exposure leads rapidly to muscle paralysis. Once inside the endocytic vesicle, the acidic environment triggers a critical conformational change in the heavy chain. The low pH causes the heavy chain to insert itself into the vesicle membrane, forming a channel. This channel acts as a conduit, allowing the light chain to escape the vesicle and enter the neuronal cytoplasm, or cytosol. This process, known as translocation, is essential for the toxin's activity. If the light chain remains trapped within the vesicle, the toxin is inert. The heavy chain thus serves not only as the delivery vehicle, but also as the mechanism for cytoplasmic release. The light chain, now free in the cytosol, functions as a highly specific protease. Botulinum toxin exists in 7 distinct serotypes labeled A through G, each cleaving a different target protein or site. Serotypes A, C, and E target SNAP25, a protein crucial for vesicle docking. Serotypes B, D, F, and G target VAMP, also known as synaptopepin, or syntaxin. These target proteins are collectively known as SNARE proteins, which stands for soluble N-ethylmaleimide sensitive factor attachment protein receptors. The light chain requires zinc as a cofactor to execute this proteolytic activity. The SNARE complex is the molecular machinery responsible for fusing synaptic vesicles, which contain acetylcholine, with the presynaptic membrane. This fusion is necessary for neurotransmitter release. The complex typically consists of VAMP, SNAP25, and syntaxin. By cleaving one of these components, the botulinum toxin light chain effectively dismantles the fusion apparatus. For example, BONT type A cleaves SNAP25 near its carboxyl terminus, preventing the formation of a stable, functional SNARE complex. This disruption immediately halts the calcium-dependent exocytosis of acetylcholine. The immediate consequence of SNARE complex cleavage is the complete inhibition of acetylcholine release into the synaptic cleft. Since acetylcholine is the primary excitatory neurotransmitter at the neuromuscular junction, its absence prevents the muscle fiber from receiving the signal to contract. This results in flaccid paralysis of the affected muscle. This state is often described as chemical or functional denervation, as the nerve terminal remains structurally intact but is rendered incapable of communication. The duration of this paralysis is determined by the time required for the neuron to synthesize new SNARE proteins and re-establish functional connections. The 7 serotypes exhibit varying potencies and durations of action due to their distinct cleavage sites and stability. Botulinum toxin type A, marketed under various trade names, is the most commonly used clinically because it provides the longest duration of effect, typically 3 to 6 months. Type B offers a shorter duration. In therapeutic applications, the toxin is injected locally in minute, highly diluted quantities. This localized administration ensures that only the targeted muscle groups are affected, minimizing systemic exposure and toxicity. Clinical uses span from treating muscle spasms like cervical dystonia to manage chronic migraine. Following intramuscular injection, the toxin is rapidly internalized by the target neurons. Systemic absorption of therapeutic doses is negligible, ensuring a high safety profile when administered correctly. The effects are not permanent because the nerve terminal eventually recovers function. Recovery involves 2 main processes: the degradation of the inactive light chain within the cytosol, and more importantly, the sprouting of new nerve terminals or the regeneration of the cleaved SNARE proteins. This regeneration process dictates the clinical duration of action, typically requiring several months. Pharmacologically, botulinum toxin represents a highly specific biological drug targeting protein-protein interactions. Its mechanism provides a model for developing other targeted therapeutics that interfere with specific cellular signaling pathways.
Research focuses on engineering modified toxins with altered receptor specificity to target non-cholinergic neurons, potentially treating conditions like chronic pain or specific autonomic disorders without causing muscle paralysis. Understanding the precise molecular interaction between the light chain and the SNARE complex remains crucial for designing next-generation neurotoxin-based drugs with enhanced therapeutic windows.